So yeah, please plan to stay until around noon time. So 2 hours time commitment we are talking about here. Okay. All right. Okay. So let's take 10 minutes break and we'll meet again.
Okay.
Bye.
Bye. See you.
Hi. Good morning for you. I am Carlos and I want to discuss about meeting symposium organized in Catania.
Kamalo, can you wait for 8 more minutes? We said 10:10. Let's let's wait 8 more minutes, okay?
I speak quickly. The problem we want to organize a symposium on amyloid in Catania.
Do you know when they actually let the people in?
Already 87 people now.
They can see your slide with 87. Just remember to unmute yourself.
Unmuted.
Hi, Carmelo. We have one more minute just before we start, and you will go first. And after that, I'll continue with introducing the speakers.
Okay.
All right, everybody, it's about time to start. Good day, good morning, and good afternoon. Very nice seeing you here again. Before we start with our seminar series, I would give Carmelo has to do a couple of announcements. So Carmelo, it's your turn.
I can speak. Okay, with Rams and Danilo Miladi, we want to organize a symposium probably this year, but it depends on the COVID situation in Europe. We want to organize, we think, in October 2021. But it's important to understand what European Commission want to say about the caution to COVID situation. I heard yesterday on the TV that the president of the European Parliament said that about from I don't know, about 15 days. She said that the European people have a special passport for COVID. Probably UK and the USA country will be free to go in Europe, but I don't know about China, Japan and Korea. Sure, from Brazil and South Africa is impossible to go in Europe, but at now is not sure what the Commission decide about this. Actually, the uncertainty is very, very large. We can discuss with Ramet about to organize this symposium in Catania at beginning of October. But the uncertainty is very, very large for this reason. I don't know. Where is Rams?
So Rams has internet troubles. So I'll guess the best thing to do is just we will keep on updating everybody about the exact time of the symposium.
Oh, we had a list of potential invited speakers from UK, Germany, France, Japan, Korea, United States, of course. Probably there is no problem because the vaccination, the problem will be how go the vaccination campaign in Europe in next month.
So let's touch base again in about a couple of weeks when we will know more. But the conference will be coming. So right for now it stays. It's happening, Carmelo, and
we need some other additional times to.
Yeah. I think it's, when we will be a more clear idea about what European Commission want to make about this situation, we can decide. I think from about our other 15 days is good to decide if yes or not, or
Okay.
move our meeting next year. I don't know, but I think 15 days is good. A good date to decide about our meeting, I think.
Yeah. Okay, that sounds like a plan. Now we'll update Rams, and I'll need to start recording. We can move with today's speakers. But before we introduce today's speakers, I just want to remind everybody about the early career symposiums, which will be coming in June 23rd and July 14th. This is on Wednesdays from 9 to 12. So please mark your calendars with that. And without further ado, I'm really glad that Dr. Michael Goddard will be speaking Today, he's a professor in MRC Laboratory of Molecular Biology, Cambridge, UK, and he has done really groundbreaking work in studying the structures of fibrils, which are straight isolated from tissues. And I'll just give it to you. Thank you so much for coming. And sorry about the delay.
No problem. Thank you very much, Magda, for your kind introduction and for inviting me. And good morning and good afternoon to everybody. And I'm going to give an overview of our recent work on the structures of tau and alpha-synuclein filaments from human brain. But most of the work that I'm going to present is on tau assemblies. So I'll mention alpha-synuclein towards the end. But I want to start with this slide here, which shows you a list of human neurodegenerative diseases, and they make up something like 90 or 95% of all cases of late onset neurodegenerative disease, and they share the presence of abundant filamentous assemblies in brain cells. Now, Alzheimer's disease is the most common of these diseases, and it is defined two types of assemblies, the so-called plaques and tangles, the other diseases by only one, and assemblies made of the same proteins are found in multiple diseases. So tau assemblies shown here in blue are found in 7 of these 13 diseases. Alpha-synuclein assemblies shown in red, another 3. And filamentous tau forms the tangles of Alzheimer's disease. Now, most cases of disease are sporadic, but some are inherited in a dominant manner. Huntington's disease is always inherited, whereas chronic traumatic encephalopathy is always environmentally induced. And importantly, close links exist between protein assembly and the causes of inherited forms of disease. And at the moment, there are no approved mechanism-based therapies for any of these diseases. And my talk is mostly about tau and alpha-synuclein. So in tau and synuclein proteinopathies, the normally soluble tau protein and alpha-synuclein become insoluble and filamentous. And the causes of neurodegeneration probably lie along these pathological pathways, assuming that these pathways are linear. And to better understand the underlying mechanisms, we are characterizing tau and alpha-synuclein filaments from human brain using electron cryomicroscopy, also known as cryo-EM. Now, in normal adult human brain, 6 tau isoforms are expressed from a single gene by alternative messenger RNA splicing. They differ by the presence or absence of 3 inserts shown here in red, green, and yellow. I'm not going to say much more about the red and green inserts, but the yellow insert is important in terms of neurodegenerative diseases. It encodes a 31 amino acid repeat, and depending on its presence, one can divide tau isoforms into 2 groups. There are those with 3 repeats each, and those with 4 repeats each. The repeats and some adjoining sequences make up the microtubule binding domains of soluble tau. And the known physiological functions of tau are to stabilize and bundle microtubules, as well as to promote microtubule assembly. Its expression is developmentally regulated, with only the shortest isoform, so this one here, being expressed in fetal brain. And the repeat region of tau also forms the core of the insoluble filaments, indicating that physiological function and pathological assembly are probably mutually exclusive. This slide shows a cryo-EM reconstruction of microtubule-bound tau. This is a normal protein. So tau constructs with truncated NNC termini. And tau, shown here in red, appears as a narrow, nearly continuous stretch of density along protofilaments. And alpha-tubulin is shown here in green and beta-tubulin in blue. So this is the normal sort of view of what the protein normally does in brain. However, in the diseases, the soluble protein assembles into insoluble filaments. And downstream of assembly, propagation and neurodegeneration are at least 2 things that are observed. And small tau filaments are the most active species in experimental models of propagation, whereas the molecular species of assembled tau responsible for neurodegeneration remain to be identified. I mean, they could be filaments as well, but they don't have to be filaments. If you look at human diseases by light microscopy, you find abundant tau inclusions in different shapes in nerve cells. And in some of these diseases, especially progressive supranuclear palsy and corticobasal degeneration, you also see abundant glial cell inclusions in addition to the abundant neuronal inclusion. So this is light microscopy. If you then look by electron microscopy, you see abundant filaments that are found. And this slide shows the paired helical and straight filaments of Alzheimer's disease. And each tau filament is made of at least two parts, the so-called core and the fuzzy coat. The core is basically what is required for a filament to look like a filament under the electron microscope, with the unstructured fuzzy coat being made of the rest of the tau sequence. And the fuzzy coat is a black material, and we can see that here, lining the filament on each side. So the material between these two black arrows. Now, this is all sort of at the level of, if you like, product analysis. So it doesn't tell you anything about the relevance or not of these tau assemblies for the neurodegenerative process. And for that, you need human genetics. And this happened about, 1998, 23 years ago. The identification of mutations in the tau gene that cause frontotemporal dementia and Parkinsonism proved that this function of tau protein is sufficient to cause neurodegeneration and dementia. And missense, deletion, and intronic mutations have been found. They're all dominantly inherited. And this slide shows you the sort of the current picture with 55 coding region, 10 intronic mutations that are now, so a total of 65. And as you can see, the mutations cluster in this region here. And this region corresponds to the repeats of tau. So the distal part of exon 9 is repeat 1, this is repeat 2, this is repeat 3, and this is repeat 4. There are some mutations also in the sequence after repeat 4. And in every single case with a mutation that has been looked at so far, you find abundant filamentous tau inclusions in the brains of these individuals. And depending on the mutations, these inclusions are found in nerve cells or in both nerve cells and glial cells. And the tau filaments are made of either 3 repeat tau, 4 repeat tau, or all 6 tau isoforms. And like inherited forms of disease, the so-called sporadic tau proteinopathies can also be divided into at least 3 groups, depending on which tau isoforms are found in the insoluble filaments. All 6 tau isoforms are found in Alzheimer's disease, primary age-related tauopathy, and chronic traumatic encephalopathy. 3 repeat tau is found in Pick's disease, whereas only 4 repeat tau is found in corticobasal degeneration, progressive supranuclear palsy, globular glial tauopathy, and argiophilic grain disease. And I'm going to discuss the structures of tau filaments of the diseases highlighted here in pink, which we have determined over the past 4 years by cryo-EM, and we're currently working on the structures of these 3, but haven't finished that work yet. But before doing so, I want to put this work into perspective. So in 1963, Michael Goddard in London described the paired helical filaments of Alzheimer's disease. In 1988, we showed that tau is an integral component of these filaments. And we also showed that the microtubule binding repeats of tau form the filament core, with the rest of the tau sequence forming the fuzzy coat. And in 1991, Tony Crowther used electron microscopy and image reconstruction to show that paired helical filaments are made of 2 identical C-shaped protofilaments. Now, the resolution at the time was something like 20 to 30 angstroms, which was not enough to identify individual amino acids. In 2017, we used cryo-EM to determine the structure of paired helical filaments from Alzheimer's disease brain at 3 angstrom resolution. And like Crowther, we found 2 identical C-shaped protofilaments, shown here in blue. And each protofilament is 79 amino acids long and comprises 8 beta strands. 73 of these amino acids were identified in 2017. The other 6 have been identified subsequently in the core. A tau protofilament consists of the C-termini of repeats 1 and 2, only 1 or 2 amino acids right from the terminus, the whole of repeats 3 and 4, and 10 to 12 amino acids after repeat 4. And this and the work that follows was done in close collaboration with Schoss Sherris from the Structural Studies Division of the Laboratory of Molecular Biology. Now, this shows you the 2 types of tau filaments that you find in Alzheimer's disease brain. About 80% of the filaments are paired helical filaments. So that basically the structure you saw in the previous slide. About 20% of the filaments are so-called straight filaments, which are shown here in green. And these 2 types of filaments are ultrastructural polymorphs in that they are made of 2 identical protofilaments that are linked in different ways. And the next slide illustrates this. So it shows an overlay of the 2 protofilaments from paired helical and straight filaments, showing that they share a common protofilament core. And this slide shows the high-resolution structure of a tau protofilament from Alzheimer's disease brain. 5 of the 8 beta strands give rise to 2 antiparallel beta sheets. So they are here and here. And the remaining 3 beta strands, so this is beta 4, beta 5, and beta 6, they form a so-called beta helix. And the widely used K18 and K19 fragments of recombinant tau, they end at a glutamic acid residue 372. And so they're filament structures which are known at present, but they cannot be the same as those from Alzheimer's disease brain. And this shows you the Alzheimer fold amino acid wise, consisting of the C-termini of repeats 1 and 2, the whole of repeats 3 and 4, and something like 10 to 12 amino acids after repeat 4. Now, it is possible that additional sequences from the carboxy termini of repeats 1 and 2 are found in the filament core. We haven't seen them so far, but we can't exclude that. And this work basically explains why all 6 tau isoforms are present in paired helical and straight filaments of Alzheimer's disease. More recently, we also determined the cryo-EM structures of tau filaments from posterior cortical atrophy, which is a subtype of Alzheimer's disease, and from primary age-related tauopathy. And we showed that those filaments are exactly the same as those of tau filaments from Alzheimer's disease by cryo-EM. Now, 13% of filaments from this case here, primary age-related tauopathy, case 1, were chronic traumatic encephalopathy type 1 tau filaments. I'll come to those in a minute. Now, this individual had undergone severe head trauma during life, which probably explains the presence of these filaments. What this work also shows, the one on primary age-related tauopathy, is that the Alzheimer tau fold can form in the absence of beta amyloid deposits, because that's how a part is defined. The next slide shows you the work we've done on chronic traumatic encephalopathy. So this is another disease where you have tau filaments made of all 6 tau isoforms, and it is associated with repetitive head impacts or exposure to blast waves. It was first described as a so-called punch-drunk syndrome, dementia pugilistica in retired boxers about 100 or so years ago. And chronic traumatic encephalopathy has since been identified in former participants of other contact sports, ex-military personnel, and after physical abuse. It is defined by an abundance of hyperphosphorylated tau in neurons, astrocytes, and cell processes around blood vessels. And this, together with the accumulation of tau inclusions, cortical layers 2 and 3, distinguishes CTE histologically from Alzheimer's disease and other tau proteinopathies. However, until the present work, the morphologies of tau filaments from CTE were unknown, and we determined the structures of tau filaments from the brains of 2 ex-boxers and one former American football player, all with CTE clinically. And filament structures were identical in all three cases, but they were distinct from those of Alzheimer's disease. Now, similar to Alzheimer's disease, all 6 tau isoforms assemble into filaments in CTE, and residues 274 to 379 of 3-repeat tau and 305 to 379 of 4-repeat tau form the ordered core of 2 identical C-shaped protofilaments. However, a different conformation of the beta helix region creates a hydrophobic cavity that is absent in tau filaments from Alzheimer's disease. And this cavity encloses an additional density that is not connected to tau, which suggests that the incorporation of cofactors may have a role in tau aggregation in chronic traumatic encephalopathy. And we distinguish between type 1, this one here, and type 2 CTE tau filaments. Over 90% of filaments are of this type, so type 1, and about 10%, well, about 7% of filaments in these cases were of type 2, and the other 3% were helical filaments of the Alzheimer type. Now, type 1 and type 2 chronic traumatic encephalopathy filaments are again ultrastructural polymorphs, and they have distinct protofilament interfaces compared to those of Alzheimer's disease. So there's one protofilament in chronic traumatic encephalopathy, as there is one in Alzheimer's disease, but the 2 types of protofilaments are different in structure. And this shows you a schematic of the chronic traumatic encephalopathy tau filament fold. The hydrophobic cavity and the additional density are shown. So this is the additional density here, and this is the hydrophobic cavity. And the next slide now compares the chronic traumatic encephalopathy and Alzheimer tau filament folds, and the positions of serine 356 and leucine 357 in the 2 folds are highlighted. In B here, you can see an overlay of the chronic traumatic encephalopathy and Alzheimer folds, and in C, the distances between the carbon atoms of leucine 344 and isoleucine 354 are shown as orange dashed lines. And panel D shows a view normal to the helical axis of the chronic traumatic encephalopathy fold beta helix, depicted as 3 rounds. And so they are these 2 structures are made of the same tau isoforms, but they differ in detail. Although overall they are quite similar. They're identical in this region here, but the difference is basically at the level of the beta helix. Now, in Pick's disease, which is a frontotemporal dementia with abundant 3-repeat tau filaments, we found 2 types of filament, which we called narrow and wide Pick filaments. And this is again from human brains, individuals who died with Pick's disease. Narrow filaments, which make up about 90% of filaments, are made of 1 protofilament, whereas 2 identical protofilaments are present in wide Pick filaments. Residues 254 to 378 make up the Pick tau fold, and they consist of around 2/3 of repeat 1, the whole of repeat 3, the whole of repeat 4, and 10 to 12 amino acids after repeat 4. And as expected, residues 275 to 305, repeat 2 are missing because this is a 3-repeat tau proteinopathy. And this shows you the schematic view of the Pick fold, and you can see just looking at the shape of it, it's completely different from the Alzheimer and CTE folds. This one comprises 9 beta strands, giving rise to 4 anti-parallel beta sheets. There's no beta helix, and beta strand 9 is located on the other side of beta strand 8. Another disease we looked at is corticobasal degeneration. This is now a disease where only 4-repeat tau isoforms are found in the abnormal filaments. And the filament core here extends from lysine residue 274 to glutamic acid residue 380, spanning the last residue on first repeat, the whole of repeat 2, the whole of repeat 3, the whole of repeat 4, as well as 12 amino acids after repeat 4. And all 3 cases of corticobasal degeneration that we looked at by cryo-EM had these tau filament structure, and the core adopts a previously unknown 4-layered fold, which encloses a large non-proteinaceous, probably negatively charged density shown here. And this density is surrounded by the side chains of lysine residues 290 and 294 from the second repeat, and lysine residue 370 from the sequence after repeat 4. And single and double protofilaments are found with doublets forming the majority. And here you can see a CBD filament with two of these identical protofilaments. Now, the corticobasal degeneration tau fold comprises 11 beta strands, and the internal density that is probably hydrophilic, shown again here. And since CBD filaments are made of 4-repeat tau, residues 275 to 305 of the second repeat are present, and you can see here the 4-layered structure of these filaments. So the next slide sort of summarizes what we know about the tau filament folds of Alzheimer's disease, chronic traumatic encephalopathy, Pick's disease, and corticobasal degeneration. They comprise 79, 76, 94, and 107 amino acids, and have 8, 9, and 11 beta strands, respectively. Now, each disease has its own protofilament fold, establishing the existing molecular conformers of assembled tau. Differences in tau filament structure are between diseases, they're not between individuals with a given disease. So if you look at different individuals with Alzheimer's disease, for instance, you always find this fold. Now, in chronic traumatic encephalopathy and corticobasal degeneration, internal densities of unknown composition are found, and based on the surrounding amino acids, the densities of chronic traumatic encephalopathy are probably hydrophobic, where those of corticobasal degeneration are hydrophilic. In all these diseases, external densities are also in evidence, but they're not shown here. And these external densities, so these are on the outside of the filament, they may correspond to post-translational modifications and or additional molecules. Now, filaments assembled from recombinant 3-repeat and 4-repeat tau in the presence of heparin differ in the structure, in structure from those extracted from human brain. And their cause lack repeat 4 and the sequences after repeat 4. And we show here just one example of a 4-repeat filaments, the so-called most abundant one that we found, the so-called snake filament, which accounts for something like roughly half of all filaments. But none of these recombinantly assembled tau filaments contain repeat 4 or the sequence after repeat 4. So this is at the level of protofilaments. Now, for each tau proteinopathy, we observe 2 filament types. 2 identical protofilaments can combine to give rise to filaments that are ultrastructural polymorphs because their protofilaments are linked in different ways. So the paired helical and straight filaments of Alzheimer's disease, for example, are such ultrastructural polymorphs. 2 protofilaments are always found in Alzheimer's disease and chronic traumatic encephalopathy, but in Pick's disease and corticobasal degeneration, 1 or 2 protofilaments are present. And this slide shows the amino acid sequences that make up the cause of tau filaments in these various diseases. So repeats 3 and 4, and something like 10 to 12 amino acids after repeat 4 are always present. A large part of repeat 1 is present in Pick's disease, as is the whole of repeat 2 in corticobasal degeneration. And more recently, we've also looked at the possibility of determining the binding sites of small molecular ligands, for instance, PET ligands to tau filaments, so far from Alzheimer's disease. So we looked at this PET ligand here. It is also known as APN or aprinoia 1607, also known as PBB3. This is the chemical formula, and you can see here a PET image on a control and an Alzheimer's disease patient. Now, this slide shows cryo-EM maps in the presence and absence of APN 1607 using Alzheimer filaments. And the difference maps identified binding sites in the beta helices of paired helical and straight filaments between residues arginine 349, glutamine 351, which is the so-called site 1, and between glutamine 351 and lysine 353, the sites 2A and 2B. Now, these sites run parallel to the long axis of the filaments, and the third major site that runs perpendicular to the filament axis was only found in straight filaments, which is the one shown here. And this slide shows you top and side views of the extra densities in the AP 1607 binding sites of paired helical and straight filament maps. And on the right, you can see the relative densities at binding sites 1, 2A, 2B, and 3. And this work shows that it is possible to determine binding sites of small molecules in these amyloid filaments by cryo-EM. And we hope that this work will lead to the development of more specific and sensitive PET ligands for assembled tau. Now, this is all I'm going to say about tau. Just want to spend a few minutes talking about alpha-synuclein. So basically, everything shown now done for tau, in principle, you can do for other assembled proteins, so alpha-synuclein, beta-amyloid, TDP43, and so on. And we've done some work on alpha-synuclein. Now, unlike tau, alpha-synuclein is simpler in a way because it exists as a single protein of 140 amino acids, and disease-causing mutations are located in the amino terminal half. They're not shown here, but duplications and triplications of the alpha-synuclein gene can also give rise to familial Parkinson's disease. So simple overexpression of the wild-type protein can give rise to disease. And synuclein proteinopathy, there are three major ones: Parkinson's disease, dementia with Lewy bodies, and multiple system atrophy. And we determine the cryo-EM structures of alpha-synuclein filaments from multiple system atrophy, which we published last year. And so here what we found is 4 different protofilaments that give rise to 2 filament types. And the non-proteinaceous, probably negatively charged density is present at the interface of these protofilaments. So you see that here and here as well. And the protofilament interfaces are much more extensive than in tau filaments. And as for assembled tau, these structures are different from those of assembled recombinant alpha-synuclein. And we're in the process of determining the 3-dimensional cryo-EM structures of alpha-synuclein filaments from Parkinson's disease and dementia with Lewy bodies. Based on 2-dimensional class averaging, these structures are probably going to be different from those of multiple system atrophy. And in conclusion, I would say that this work has shown that the ordered cause of tau filaments from Alzheimer's disease, chronic traumatic encephalopathy, Pick's disease, and corticobasal degeneration adopt distinct folds, establishing the existence of molecular conformers of assembled tau. And for a given disease, different individuals have the same tau filament structures, at least so far. Tau filaments are made of 1 or 2 identical protofilaments, and the protofilament interfaces are short. And ordered cores of type 1 and type 2 alpha-synuclein filaments from multiple system atrophy are made of 4 different protofilaments with extended interfaces. So this is quite different from what we've seen for tau filaments. And the cryo-EM structures of tau alpha-synuclein filaments from human brain differ from those of assembled recombinant proteins. And the major aim, obviously, for the future is to get recombinant proteins to assemble into structures that are like those from human brain, if one really wants to understand what's going on. And cryo-EM can be used to identify the binding sites of small molecule PET ligands in amyloid filaments. And finally, I would like to thank my colleagues and collaborators, and especially Shaw's sharers from the MRC Laboratory of Molecular Biology, with whom the cryo-EM work was done. Thank you very much.
Thank you so much for the great and exciting talk. We are going to have 10 minutes questions for you from the Q&As, and I'm going to read the questions for you if you don't mind. And I'll start first with anonymous question from anonymous participant. And after that, Marcus, if you don't mind, you will continue with your questions. You have a bunch of them. That's why I let you into the panel. So the first question, is it known which isoform is the most abundant one?
Yeah, so if you look in normal human brain, you find that the 3-repeat and the 4-repeat tau isoforms are present in roughly the same amounts. If you look at individual isoforms, I think the most abundant one is probably the 4-repeat tau isoform with 1 N-terminal extension. The least abundant ones are the longest 3-repeat and 4-repeat tau isoforms, so those with 2 amino-terminal inserts. What was very surprising, when the first mutations in the tau gene were identified, was that the intronic mutations that cause frontotemporal dementia, all they do probably is they change the ratio of 3 to 4-repeat tau isoforms, so that there's relatively more 4-repeat tau made than 3-repeat tau, and this appears to be sufficient to cause frontotemporal dementia by age 50.
Thank you. Marcus, it's your turn now.
Okay, thanks, Michel, for fantastic work. So I have a couple of questions in principle, but so I was always wondering how you bring together that the ALZ50 and MC1 epitopes antibodies require the N-terminal epitope, but in all the structures, this part is not seen. So how can you bring together? This is my first question. Yeah, I have more, but
yeah, I mean, we in the original paper on Alzheimer's disease we published, we see less well resolved densities, and we interpreted for the paired helical filaments on the outside and for the straight filaments at the interface between the two protofilaments, and we interpreted those as possibly corresponding to residue 7 to 9 of tau. So this is the EFE sequence, which is part of the ALZ50 and MC1 epitopes. But it remains to be shown, though, more firmly that this is really what's happening. It is true, I think, that well, certainly in Alzheimer filaments, it's true that these antibodies see the filaments. I'm not so sure. I can't remember now whether they also see the filaments in these other diseases or not. We certainly haven't observed anything in the structures from those diseases that we could interpret in this way.
Yeah. So my next question would have been for CBD, if the ALZ50 and MC1 recognizes the fibrils there.
Yeah, I'm afraid I should know, but I don't know. Yes. Yeah.
And then my second question would be, for the Pick's disease patient, do you know if the patient had any genetic mutation?
Yeah, so a particular one we looked at in tau did not.
Okay,
but there is a mutation in the tau gene, which is the delta K280 mutation. So it lacks that residue, and probably the effect of that mutation is at the splicing level. So there's relatively more 3-repeat and 4-repeat tau made. And so the 3-repeat tau that would be deposited is wild type because the mutation is in the extra repeat. And those people that have been looked at with that mutation have what looks like PIC bodies. And we're in the process of doing the cryo-EM structures from those to see whether they're the same or not.
Thank you. If I may, one last one. Your last comment you made with respect to the Parkinson's disease and DLP fibrils. So did the situation change? So they are not fully straight anymore, such that one can do the cryo-EM, or?
Well, we're working on it, and the reason why we haven't got it yet is because they're pretty straight. So I can't say really, but they certainly, just at a negative stain level, they look different from the MSA ones. But no, we don't have any structure at the moment.
Thank you so much.
So I'm going to continue with reading the questions. There's a couple of them which are kind of similar. I'll read the one from Amir, which is kind of the similar. He says, great talk. Have you find functional distinctions such as toxicity or seeding potential among these various style of filaments? And I'll continue with the other questions. So,
yeah. So, well, toxicity, I have no idea. I mean, well, depends what you mean by toxicity. I don't know. Seeding potential, yes. I mean, if you take seeds from Pick's disease, for instance, and you add, so they are made of only 3 repeat tau isoforms, and you add a sort of recombinant 4 repeat tau, you get no seeded assembly. But when you add recombinant 3 repeat tau, you do. And for corticobasal degeneration, it's the other way around. Now, for Alzheimer's disease, which is interesting, is that the core of the filaments, almost all of it, so the residues 306 to 380 or so, are common to 3 repeat and 4 repeat tau isoforms. But you do find all 6 tau isoforms in the filaments because you have the carboxy terminal ends of repeats 1 and 2 also in the core, but there's only so far as one, perhaps 2 amino acids. So in principle, it could be possible, I think, to form paired helical filaments from either 3 repeat tau or 4 repeat tau, but this hasn't been shown yet. In terms of the seeded assemblies, you can do these types of experiments using Alzheimer's disease seeds, but whether the structures of the seeded assemblies are the same as those of the seeds, I think that remains to be seen for all of these diseases. We did some work on alpha-synuclein using seeds from multiple system atrophy brains that we have the structures of, and we added recombinant alpha-synuclein to those, and the seeded assembly structures are different from those of the seeds. But of course, this alpha-synuclein is just recombinant, it's not modified in any way. So that's another thing I think for the future is to determine what is required for a seeded assembly to look like a seed structurally.
Thank you. So I'll continue with another question from Amir. He's also wondering if the structure of heparin-induced tau fibrils are changing by altering the agitation, temperature buffer, etc.
Well, I mean, all I can say at the moment is that if you use recombinant protein, probably we may not have used the right conditions, but all the conditions we tried where you form filaments from recombinant protein, the structures are different from those you find in human brain. So I think that's something for the future. It's obviously, it should be possible to form filaments with the same sorts of structures, but at the moment we haven't succeeded in doing that.
So yeah, there's the following question is also down these lines, but it's kind of interesting to hear your answer too. It's from David Elizer. And he says that you had a recent paper describing in vitro seeding synuclein fibrils using MSA fibrils, which you did not replicate the disease structure. You mentioned about that. And the question is, are you exploring other conditions to see if you can get them, just kind of turning it around using different conditions?
Yes, we are. We're trying, but both for tau and alpha-synuclein, but so far we haven't succeeded. So yeah, yes.
Right. Thank you. So we have a question from Christian Grissinger. He says, "Hi, Michael. Very nice talk. Do you have further information and additional non-protein density in CT tau filaments?" And there is a B part to the question. Is the difference in the structures just determined from a 4R, 3R, or are there any additional factors determining the fault, which is you're getting these fibrils from a very complex tissue? So yeah.
Yes. So hi, Christian. We don't really know. I mean, all we know is that in Alzheimer's disease, we don't have this density, and the beta helix is different, and in CTE we do. But of course, we do not know whether the fold is different and then the density comes in, or whether the density somehow is responsible for determining the fold. I think that's an important question. I mean, if you think about CTE as a disease, it's something, well, it's about as non-specific as you can get. You know, it's bashing somebody on the head repeatedly. And then 20 years later, the person can get this quite specific condition. But it's also true that when the injury happens, probably lots of, I don't know, axons, for instance, are damaged. They contain lots of lipids, and maybe one of these, small lipids is this sort of hydrophobic density we see. At the moment, we're trying to find out what it's made of. I think that's probably what one has to find first using mass spectrometry. And for corticobasal degeneration, of course, it's a similar sort of question, whether the differences in isoforms are all that is needed to determine the differences in structure. I do not know, but as I said before, I think in principle it could be possible to form paired helical filaments just from 4-repeat tau or just from 3-repeat tau. So it may not just be the isoform composition.
Thank you very much. For the sake of time, and we will give the next speaker time to talk. If you don't mind, Michael, staying around, there's so many questions, and you can browse through them to select.
I'll have a look. Yeah.
I'm going to be checking on them just to group them by meaning, and we are going ahead with our next speaker, Bingzu. Bingzu, do you mind starting sharing your screen? And it is my pleasure to introduce you and have you around. Bingzu is an assistant professor in North Carolina Central University, USA, and he has been a very active participant in our ZOOMinar series. Thank you so much for your insightful questions. We all enjoyed, and it's really great to have the opportunity now to hear about your research. Thank you very much for coming.
Great. Magna, can you see my slide?
Yes, yes.
Okay, wonderful. Hi, everybody. Greeting from the Research Triangle, North Carolina, United States. My name is Bingzu. I'm going to talk about from a different approach, which is from Michael's powerful structural biology. We're going to approach this protein neurodegenerative, the protein misfolding problems with small molecule library screening approaches. And hopefully I'll give some example as a part of proof of concept ideas. So talking about the pandemic, we also have two less visible pandemic going on for decades. So one is a global diabetic pandemic, and the other is Alzheimer's disease. So I think I probably don't need to mention a lot of those details. Once that maybe just a few points is there are over 300 million people have diabetes worldwide, of course, largest in China, India, those big nations. And the worrisome trend is half the people who have diabetes do not know they have the disease already. Once they know, it's already too late because they are irreversible. And also another worrying trend is half the people who die from diabetes, they are under the age of 60. So it's getting younger and younger. Roughly about 10% of the population in the United States and in Europe have a diabetic problem, and which impose a huge economic burden to the society. Similarly, regarding Alzheimer's disease epidemic, more than like 5 million Americans or Europeans, they live with a disease, and this number expect to double or triple in the next few decades. And with the technology development, some of the major diseases, such as heart diseases, the death rate is actually decreases over time. However, for Alzheimer's, this is dramatically increased since 2000. So these are should getting urgent attention for these two epidemic. Now, interrelated to those two diseases, one of the feature is a protein amyloid and so-called protein misfolding diseases. And there are, so far, there are over dozens of protein misfolding diseases has been discovered and described. And here is some of these are showing here, and some major portion are related to neurodegenerative diseases such as Alzheimer's, Parkinson's, and prion disease, as well as a significant portion are non-neurodegenerative disease such as related to diabetes, heart disease, or disease related to eye. And regarding the protein amyloid, I just want to say a few words. I think this audience probably don't need much introduction. This is the work from late Professor Chris Dobson from Cambridge, describing the process of amyloid development. So start from monomeric protein, then initial phases is the forming the oligomers and protofibril formation, and then leads to lateral association of protofilament, leads to mature fibrils. And both this intermediate phases, oligomers, protofibrils, and mature fibrils are to be very toxic to the cells, and the killing of nearby cell, they can lead to deposits and plaques to related tissues. And similarly, as the development of protein amyloid kinetically can be visualized using certain biophysical means, and showing here, this is a THT fluorescence measurement showing the development from the monomeric protein to oligomeric, which has beta rich structures, as well as to the mature fibril. And these oligomeric and mature fibril, they have the capability binding to certain fluorescent dyes, such as diaflavin T showing here, and then they can be monitored readily by fluorimeter. And those kind of assays are very common using for example, for drug screening. And I think they here showing the bottom showing several examples of biophysical means to monitor the amyloid formation for use to monitor their formation, such as the fluorescence-based method using circular dichroism based on the beta structure, as well as the electron microscopy visualization. So as I mentioned in the previous slide, the toxic oligomeric and other intermediate form, as well as mature fibril, they are toxic. So one of the commonly popular way is to inhibit those protein amyloids. And there are a variety of means to identify those new inhibitors. And I list a few approaches here, including high throughput approaches, such as using diverse molecular scaffold, using computational aided method, structured based screening, as well as others such as drug repurposing, and using like early on using epidemiological alternative medicine approaches. Regarding for the screening of small molecule libraries, there are a number of different kind of property of library, including early on the natural product library, usually relatively small, and you can obtain from commercial vendors. And also each lab can make like targeted small libraries based on the property you are looking for, as well as a repurposing library. For example, NIH provides some small repurposing library. Those are FDA approved drugs or investigational compounds, and it has been go through animal toxicity assays, and you can know whether they are brain penetrant or not. So which can facilitate the process and time of the compound discovery. And there are also, if you want to discover more novel compound, you can use a diversity library, which is much larger, like from 20K to 200K library, which you can screen. There are commercial sources as well as through academic collaboration. And additionally, there is a combinatorial library using the FRAC combinations, which will provide a huge number of possibility for really novel structural scaffold discovery. So regarding the screen assays, and there are here list of showing two graphically showing for the in vitro assay for screens, such as using THT fluorescence-based assay I just mentioned early on, as well as a cell-based assay. You can use an imaging or using looking at the property of the cell, such as cell viability assays for using for screening libraries. And I think more recently, I also saw in the field that are developing like animal-based screening, although these are relatively low throughput. For example, there are intelligent kits from TAC's system, which can, in each case, can screen for 16 animals. So in terms of protein amyloid inhibitors, there are, in terms of their classes and mechanism, there is a lot of research has been studied and contribute to our current understanding. And many of those names, you're probably familiar to this audience, including late Professor Chris Dobson, Professor Tony Fink from UC Santa Cruz, as well as others. And I apologize if your name is not there, you have done a lot of work, the list is really huge. But so far we know from the literature, there's lots of flavonoid, including EGC gradient from green tea, often showing as a positive control, phenolic compounds, such as the cumin, which is also well established, as well as a lot of diverse structure of a compound from a simple molecule such as a sugar like inositol, vitamin K3, to complex molecules such as molecular tweezer, and also tafamidis. This is the FDA-approved drug for treating the TTR amyloid, I think developed by Jeff Carey's group. But overall, the mechanism of inhibition is still not fully understood. So that is a need to study the mechanism and to find also new novel compounds. So I want to briefly introduce the system we are studying. We study amyloidosis related to type 2 diabetes. And this is the sequence of human amylin, as well as the rodent amylin, which has a different sequence issue, which is actually have a significance. Amylin is also called IAPP, islet amyloid polypeptide. It's a protein hormone co-secreted from insulin from pancreatic beta cell. The physiological role of amylin is most related to glycemic regulation. However, its pathology and most attention. And in the insulin resistance in type 2 diabetes patient, it demands to secrete more insulin. Therefore, it will secrete more amylin because they're co-secretion. However, hypoamylinemia, meaning high concentration of amylin in the circulation, promote toxic amyloid formation, which can be detrimental to the pancreas itself, or when it circulates, it can cause damage to other tissues. And here showing the post-mortem pancreatic tissue with the position of amylin amyloid, the same way the cytoflavin S, showing in white. And one of the interesting thing is that using in the mouse model here, the mouse transgenic mice as overexpressed human amylin actually recapitulates the human disease patient in the scenario. This is showing the white diaphragm as sustaining amyloid. However, non-transgenic control showing no such thing, suggesting the sequence specificity, which is showing like including 3 protein possibly disrupting the beta structure. More recently, the clinical research showing amylin amyloid may also deposit in the brain, because this is like from the peripheral, and also this is the staining data from the literature showing the deposition of amylin amyloid in the blood vessel of the brain or parenchyma. And this is also staining showing the A beta staining, showing in some cases they are co-localized, and in other cases they are not co-localized. Exact the interaction between those two molecules is still not known. So one of the hypotheses is maybe amylin amyloid is a new amyloid related to neurodegeneration. And one piece of supporting data is through a transgenic rat model. It's called the hip rat. And this rat model is this is a rat, and because as I mentioned earlier on, the rat sequence of amylin is not amyloidogenic. And this hip rat overexpresses human amylin, and you see a significant deposition amylin plaque in the pancreas. as well as you see some deposition in the brain of hip rats. And those rats showing neurological deficits related to learning and memory, such as in rotor rod tests, in recognition memory, as well as explorative drive as shown by rear up tests. So in our study, we start with amylin amyloid. The first thing to test whether they are cytotoxic, and we choose a variety of relevant cell lines, including the beta cell line, including neuronal cell line, SHSY5Y, Neuro2A cell line, as well as heart-related smooth muscle cell line. And in all cases, amylin amyloid showing a significant cytotoxicity compared to the control. And in comparison to A-beta-42, amylin is even more toxic under the same concentration as showing here. So I think two other players which will be mentioned in my talk as well are the two established biomarkers for Alzheimer's disease, A-beta and tau protein. I think I do not probably go even further since we just talked about, Dr. Goedert just talked about tau, and I think A-beta as well studied. So outline of my talk is we're trying to address amyloid inhibitor drug discovery, specifically what kind of a compound class, or more specifically the functional groups are important for inhibition, and what are possible mechanisms of inhibition. And I'll also provide some translational application related to those aging diseases. And I will discuss two aspects. One is related to the mechanism of inhibition, and I have several studies related to the functional groups we will discuss using one of the example of a Bakelin to relate to the mechanism, and also the role of redox as important player related to the some of the inhibitors. And I also give a short story related to rosmarinic acid using for translational application. Okay, so first we did a targeted library screen, and this is a relatively small library of about 100 compounds, and these are all natural compounds we pick in our lab based on the literature showing anti-amyloid, anti-diabetic, or neuroprotective action. And of course, there is lots of flavonoid, polyphenols in those in the library. And here is the result. This is showing x-axis is the percentage. This is fluorescence-based assay. This is a negative, which will be 100%. And some of the positive controls like EGCG of curcumin showing us as a strong inhibitors. Now, if you look at the inhibitors, and a lot of those positive inhibitors, and a significant percent, over about like half of those are containing catechol groups. And here is the catechol chemical structure in comparison a control called a phenol, which is non-active. And we did, you try the catechol itself, showing mild effect, inhibit amylin amyloid. This is a fluorescent TH assay. We thought our initial natural product-based targeted library are very biased because we choose them. Now we're going for more diverse library. This is the NH library, 700 FDA-approved drugs or investigational compounds, and they are very diverse with a very different kinds of structure template. And what we further do is, instead of just amylin, we also expand to 3 well-established amylin amyloid. Amylin, the longest isoform of tau protein 2 and 4R, and the A-beta-42. Okay, let me explain this graph showing here. The x-axis is the compound ID. So from 1 to 700, each dot represents a drug. Now the y-axis is the fluorescence in log scale. So 2 represent 100%, so it's negative. There is no activity of those compounds. If it's 1, that's 10% of intensity, so that's a significant strong inhibition. So anything like a small number close to 1 or below, those are a strong inhibitor hits. And the results of this, of course, we find dozens of hits, and showing some of those are showing red dots. And significant portion of those strong hits, in the case of amylin, about 1/3 of them. In case of a tau and A-beta, about nearly half of them are catechol-containing compounds, which is consistent from the previous screening. And here just more, looking at the chemical itself using TEM, demonstrating its inhibitory effect compared to the buffer or compared to phenol, which is showing very nice rigid long fibers. So what are these compounds? So I think we look at those the strong inhibitors, look at those hits, and as mentioned, a large percentage are chemical containing compounds are showing here, highlight the dihydroxyl group showing here, and then redox relate to hydroxyl, that is also acetylquinone type of a structure, showing there are multiple of those showing that. These are related in terms of relate to the redox. We also find other classes of compounds such as tetracyclines, which for the time being, I don't have time to go into, but I think we'll focus on these catechol-based inhibitors. So because the assay THT-based fluorescence basically occasionally can be showing false positive as shown by Dan Roy's group in early literature. So we certainly in all the hits, we do the validation with multiple secondary assay. And here showing two assays. One is called photo-induced cross-linking of unmodified protein pickup assay. And the other is amyloid remodeling assay. In this assay, this is a transition metal induced cross-linking of oligomer. So if you have a no inhibitor, that you will see a monomer, dimer, or trimer, or trimer, or pentamer. But with the inhibitor, such as the rifamycin, which is a no inhibitor, you see only monomer, that is no dimer or trimer. Okay, so by this way, we can verify, and a lot have been verified, such as rifampicin, rifampicin, doxorubicin, as well as a number of DOPA-related compounds. And similarly, this is the amyloid remodeling assay, is a fluorescent THT-based. You see the formation of a protein amyloid, and then you spike the compound, and EGCG, for example, that is a remodeling events, that is a decrease in fluorescence intensity. And this way, we can verify with some of the strong inhibitors as well as weak inhibitors. So now this is really the functional group. I think we identified the catechol containing compounds. Now, I want to give one example, which also contains a catechol compound, the bakelin as example. So bakelin is also discovered library screening, similar as EGCG and curcumin as positive control, showing a strong inhibitory effects. And bakelin is from the beautiful flowering plants, Aspergillosis. And the chemical structure of a bakelin is showing like this, the 3-hydroxyl group attached to the benzene ring. And these are the validation of a biochemical assay showing bakelin can strongly inhibit amyloid formation. This is the control. And in the remodeling assay, you're adding the bakelin, spiking, and leads to the reducing of fluorescence intensity in the THT assay. And IC50 roughly estimate to be 1 micromolar. We also did the cell-based assay. For example, this is the amylin amyloid causing cytotoxicity. And then with the addition of a bakelin, you see a reversal of the cell viability, and this is dose dependent. And the bakelin itself has no effect to the cell viability to the cells. Now, what we're interested in is based on the bakelin structure showing here, we ask the question, what functional groups are important? So looking at the structure of a bakelin, it has 3 hydroxyl groups. So we are wondering whether those 3 hydroxyl play a very important role in terms of their activity. So we change those to the ether groups, or just use substitute with like a single hydroxyl at a position of 5, 6, or 7. Or a dihydroxyflavin analog 5,6, 5,7, and 6,7, and test they are using various assays I have described in the previously. And this is the fluorescence inhibition assay. This is the amylin amyloid showing the amylin, showing the compound showing the inhibition. Now, all of this singly hydroxyflavin analog showing no activity at all, as well as the methyl ester analogs. However, dihydroxyflavin analog all showing activity, especially 5,6 and 6,7, this adjacent hydroxyl group connect to the benzene ring. And we know this is the catechol moiety of the compound. And this is also showing in amyloid remodeling assay, which has a similar result. This is the positive control of the bakery itself. And this is 6 and 8. 7 have mild activity. And additional using pickup assay and cytotoxicity assay all have a consistent result showing this dihydroxyl group are important as related to their inhibitory effects. And we further characterize whether it's possible maybe this compound has actually interact with the amylin itself, the peptide. And we did a mass spectrometry compared to the control, just amylin itself, and amylin plus bakelin showing in blue. And yes, indeed, we find this is the MS experiment showing the new peak showing up. And when we do the mass spec ion the map, it not only shows amylin itself, in this case, in both sample, you're showing the amylin peaks, +3 or +4, and they are overlap on each other. However, only the bakelin mixture, whereas amylin, you're showing the new peak of amylin added in both +3 and +4 peaks. And so this leads us to believe that showing the evidence of the conjugation of the bakelin inhibitor to peptide through the shift-based formation. And I think we verified the molecular ion mass for that. So lastly, to the mechanism, I want to mention redox status can play an important role. And this is really a serendipitous finding. My student did the experiment and leave some of the catechol containing compound just on the table of the bench and go away for a couple of days. And when he without capping the lid, and when he come back, find some of those, a lot of those catechol containing compounds actually showing significant high inhibitory effects. So in the panel A, this is a THE fluorescence assay showing the negative control of phenol, and the morin, which is an inhibitor. However, this is not catechol containing compound, so there is no difference. Now, all the other catechol containing compound, including catechol itself, showing this is a freshly made versus the brown is the exposed to the air. We call it aged. Okay, so in all these cases, most dramatically is norepinephrine using dramatic increase after auto-oxidation. This is a oxidation over 2 days, 48 hours to the air. So all these are consistent, and this is just to pick up the 2 examples. One is rosmarinic acid showing here increase whenever, and this is a dramatic increase in the case of norepinephrine. And this is also being further confirmed using off-pathway assays, such as showing this is the amylin, and once conjugate in treatment with inhibitors such as rosmarinic acid or norepinephrine, it's showing a smear of a band on the Western blot against anti-amylin, which are the SDS-resistant broad range molecular weight off-pathway species. And then this species will be reduced, will be under reducing agents such as cysteine, glutathione, or cysteamine. Those are reducing reagent. Treatment, they will be gone, after treatment, and this is a dose dependent. So, suggesting this redox is really play important role in terms of for this class of catechol containing inhibitors. And I think now we also did a more rigorous study using anaerobic chamber conditioning, and this is which is similar to the previous slides. In all the cases, aerobic conditioning is much higher inhibitory effects versus fresh inhibitors. Okay, and this is also showing in the case of norepinephrine, anaerobic conditioning, there is almost no difference, and in the aerobic conditioning, you see significant enhancement. This is a TEM study showing the anaerobic, both cases, you have the lots of fibril structure of amylin fibrils, but in aerobic, in the norepinephrine case, most of the fibrils are gone. Now we are wondering what are these auto-oxidation in terms of chemistry. So we want to identify the oxidized product of these amyloid inhibitors. This is we use norepinephrine as one case. And this is a UV study, initial UV study showing the fresh, showing the black, and then in aerobic condition, there is no change, overlap on top of each other. However, in the aerobic condition, after 4 days, you see big change in terms of the UV absorption, showing new species formation. And we did LC-MS study. In the LC, you see the new species formation, and also growing up over time. And then we use a mass spec to identify some of those species. One of those is oxidized norepinephrine called a local adrenochrome. Okay. And I mean, all these data suggesting a covalent mechanism, and which is the inhibitor of catechol. They can be oxidized and then can conjugate to the amine group of the peptide, and therefore can lead to protein inhibitor complex can block the extension of a beta stacking, therefore block amyloid formation. However, our mechanism is not, it does not exclude other mechanisms such as non-covalent interaction proposed by a number of groups in the field.
Bingzu, you have a couple more minutes.
Okay, yeah, so I'm getting to my last story for the talk, so related to the rosmarinic acid. So this is similarly, from the library screening, we find rosmarinic acid as a strong inhibitor, and this is a find from Rosemary. And here I'm showing the inhibitory effect. Half of the fibrils became non-amorphous aggregates, and then the IC50 is about 200 to 300 nanomolar, and this is the T-half extent that was increased treatment, increased concentration of rosmarinic acid. And look at the structure of rosmarinic acid. It has two parts, a caffeic acid, a salvanic acid A, and both are contain a catechol group. We're wondering whether there is an additive effect. And yes, indeed, this is the rosmarinic acid by itself strong inhibitory effects. SAA and the caffeic acid has a milder inhibitory effects. But if we put those two compounds mixed together, 1-to-1 molar ratio, they are similar as rosmarinic acid, suggesting additive effect. And this is also consistent with IC50 analysis. This is for rosmarinic acid. Caffeic acid and SAA has a right shift pharmacologically, which is weaker, and that is consistent with additive effect. And also the cytotoxicity assay showing a strong the inhibitory effects of RA, the weaker SAA and CA effect. And with collaboration with Professor Bevan's group on the simulation, what we also find, this is the using peptide of amylin 20 to 29 and showing the interaction. This is a heat map or residual interaction plot. And this is the 3 molecule, 3 peptide fragments. And if you stack on top of your beta strand, you see lots of residual interaction in blue, in the green or yellow. However, with rosmarinic acid, there is strong disruption. You see all those cross peaks are gone. And for caffeic acid and SAA, it's a partial inhibitory effect, supporting the interaction of this additive effect. And RA, those also significantly induce human amylin oligomers in the serum of hip rats. This is overexpressed human amylin, and this is a dose dependent. And also we tested in using the diabetic patient, showing the time dependent of a reduction of amyloid oligomers. Now, lastly, we want to test the effect in vivo. And this we using the hip rats and using 5% weight-to-weight dosage by diet and treat for 4 months from 5 months of age, which they start to develop pre-diabetes. And as you see, this is the white type, the hip rats showing a lot of those orange color, those are Congo red staining of amylin amyloid in the islet. And then with treatment, most of those brown color, those are gone, have some residual left. suggesting an in vivo efficacy. And similar message from the amylin antibody staining, and with the Western blot of oligomers, so reduction after treatment, and also the glucose level are reduced compared to the hip rats.
Binzu, 1 more minute.
Okay.
Sorry.
Yeah, so we also showing the eye, also showing the in vivo neuroprotective effects, and for the time being, I'm going to skip the slides. And then we also did, initial medicinal chemistry to further optimize, based on rosmarinic acid as a template, and showing better the efficacy of that. In summary, so we have using library screening approach to identify catechol-containing compounds as a broad class of amyloid inhibitor. And the structural activity relationship, they can demonstrate the key roles of these hydroxyl, the dual vicinal hydroxyl, the catechol moiety of these compounds. And we propose a covalent shift-based mechanism. And we find the redox state is very important in terms of this broad class of catechol-containing inhibitors. And we identify RA has a significant not only ex vivo or in vitro, also in vivo efficacy for anti-diabetic and anti-amyloid effects. And lastly, acknowledge for people who did the lab, mainly Dr. Lingu and Paul Wielander, these are the research faculty and the talented PhD student, and a lot of collaborators to help with this project. And without these funding, we were not possible to do this work. Thank you.
Thank you, Binzu. We're now ready for questions directed. We will have 5 minutes, a few questions for Binzu before we start the joint session with questions to both Bin and Michael. So I'm going to read a question from anonymous person, and after that, so Christian has a question for you, Binzu, though it may be partially answered already. But Christian will be next after the anonymous question. The anonymous attendee asked, just wondering whether you test the toxicity of your small molecules alone, do they have any effect on cells?
Yeah, we always test that assay. So I think I show some of the control experiment. They do not affect cell viability, but we, for each, identify the hits, we always test in cell-based assay because that's very easy to do. And all the hits we showing, and most of those, there is no toxicity. I think some of the identified compounds such as doxorubicin, there are no cell toxicity for those. Actually those are cancer killing agents.
Thank you. So before I give the turn to Christian, I just want to remind all the attendees, if they want to participate in the panel discussion, to raise their hand so we can promote them in the panel. So Christian, it's you now. It's your turn.
Okay. I had written down the question before you started talking about the oxidation, and therefore it's in a way a little bit outdated. But maybe following up on the question is on the last example with the rosmarinic acid, you showed that it is cleaved in the middle and then also oxidized. So what do you think is then the active species that does the job on the aggregation? And do you have any idea about the half-life of this molecule in I guess it was a mouse model, right?
Yeah, so very excellent question. I think we have, so what I mean here is just, it can have 2 separate parts for caffeic acid and salvinic acid. This is the ester form of both, for rosmarinic acid, and that is additive effect. We have not tested caffeic acid alone or SAA alone. We expect those are weaker inhibitors. And in terms of the in vivo, the product in mice, we have not attempted to isolate those in vivo metabolites of the treatment of RA treatment, hip rats. But I think we are still, this is still ongoing work. It's going to be a challenging work because there are multiple metabolites as described in the literature for these kind of compounds. But I think it depends on the abundance, which some of those most abundant species, maybe we can isolate that and identify them.
Okay, thanks a lot.
So there is a question from Mike Sawaya. He asks if bacillin is withdrawn from bacillin dissolved amylin fibrils, and the amylin fibrils regrow.
Magda, I do not understand the question. Can you repeat? Magda, you're on mute.
Oh, sorry. Do you want to read the question one more time?
Yes, please.
If bacillin is withdrawn from bacillin dissolved amylin fibrils, can the amylin fibrils regrow?
We have not tested that. That's an interesting question. So we did test bacillin with amylin at the beginning of the time point and in the middle of the time point. And once the amylin form mature fiber, we have tested those. And of course, I think, let me see. So for example, here we tested at the beginning, so that is no fluorescence signal. And in the middle, it drops. And then once we're assuming there's a mature fiber formed because it has a fast kinetics, and that is also remodeling happening. And we have not tested the withdrawal scenario, but that's an interesting question. We can test.
Thank you very much. And now I'm going to move on the general questions addressed to both of you. And AJ has joined us, the panel, for quite a while. He raised his hand quite a while. So AJ, it's your turn to ask your questions. AJ?
Oh, it's me. Hello.
Sunil, you can go ahead. AJ is.
I'm sorry.
Okay.
Yeah,
go ahead, Sunil.
Okay, great. I have a question with to Benju. These covalent inhibitors you're talking about, they generally what happens is, let me lower my hand, I guess. Yes. So generally they, in general, covalent inhibitors, they are designed like to initially get affinity with the target, and then they enhance the affinity by binding covalently with the target. So I was wondering here, what is the mechanism of binding and the efficacy? Because if initially there is the total dependence of the activity comes from the covalent inhibition, then it could cause a problem in specificity, and could cause toxicity with other proteins as well. And the second thing is, how is the covalent inhibition, the mechanism here? Would they interact with some the monomeric state or some pre-fibril state where they can reverse the mechanism of toxicity? So those are a couple of questions I have.
Thank you very much, Neil. These are very good questions. So we know, this covalent mechanism we propose, this is just some piece of evidence showing that, supporting that mechanism. And there are other mechanisms has been proposed, as I mentioned, like the Enrollis group showing a lot of, the hydrophobic interactions or charge-charge interaction in some cases. So they are not mutually exclusive. And I think probably all these mechanisms play some role in terms of inhibitory effects. Regarding the exact mechanism, I think we still don't know. I think because of a complexity of these heterogeneity of those intermediates, but I think there are some of the powerful mass spec, like ion mobility. I think the following is a speaker will talk about that in the future, which may be able to help us to really identify, those specific species of, the conjugation or those, in more detail.
Okay. Just the relevant question, I guess. So do you like when these covalent inhibitors, I guess you adding it at the beginning or somewhere? So it would be helpful, I guess, to see the effect of these covalent inhibitors, at least in terms of where they are interacting more, or if they have any efficacy to any state. So if you have this whole process going on and you add at the beginning, middle, and at the end, and then see, maybe run a mass spec or something in order to see where they are forming these adducts, that could, at least in part, can tell you which one is more effective sites or more effective interactions of these covalent inhibitors with the species, I assume.
Yes, I think exactly. Very good question. I think we have, in terms of, we did some of the studies. We are still ongoing with animal study. For example, we can, for example, for this diabetic amylin, so we can put the treatment early on. This is for prevention. For example, when the amyloid has not been formed, or already mature, the fibrils has been formed in the late stage of the diabetes. So we see whether there is a therapeutic effects. So for those kind of those, we have done some of the in vitro experiment, but I think one thing is we'll try to maybe to find some collaborator, have some high resolution approach, so that we can really see all those intermediates and in high resolution very more clearly.
Okay. Thank you.
So I see that Rakesh, you're online, your turn, and after that, Christian, I saw that you have a question to Michael, so you will be after Rakesh.
I have two small questions to Michael. Great talk and great work as usual. So what about Brad Hyman's work about the soluble high molecular aggregates in Alzheimer's disease? So 32 cases shows differences, while the filaments in Alzheimer's are the same between patients.
Yes. Well, I don't know. I mean, I would suspect that the soluble high molecular weight tau aggregates are filamentous, and that there are differences in amounts between different people, but that the protofilament structures are the same. That's what I would think. Now, people often say that filaments are insoluble. Insoluble means if you spin something down in the ultracentrifuge, it's in the pellet, I guess. But it's been shown that if you take tau filaments, for instance, that are insoluble, you sonicate them, and then you spin them again, they stay in the supernet. So filaments can be so-called soluble. So that's what I would think. But of course, I don't know. I mean, I have no proof of this. But we've never found any differences in structures between tau filaments of different individuals with a given disease.
We did not see also with oligomers the same, like out of 15 cases, the 12 were almost the same. But the other question, I don't know if you did it, I'm not sure. Do you expect there will be different structures between different brain regions or no?
Yeah, so strictly speaking, I don't know, because we haven't, the only thing that we have done is look in Alzheimer's disease, that's frontal cortex, look in posterior cortical atrophy, that's occipital cortex, and looked in primary age-related tauopathy, that was the hippocampus. And the structures were always the same. We've done some work, but that's not using cryo-EM, that's using immuno-EM, looking at different brain regions in Alzheimer's disease, and that's indirect, okay? And based on that, it's all consistent with the structures being the same in different regions. But the caveat here is that if you use the same methods and the same antibodies on chronic traumatic encephalopathy, you would also conclude that that's the same as Alzheimer's disease, and by cryo-EM it's not. So there's a slight caveat there, but I would suspect that the structures would be the same in different brain regions.
Thank you. The final point, so assuming they are the same in different regions, this supports that they they started in one region, then they spread and seeded, or they are localized? Like,
Yes.
like as brain injury, mainly they form localized where the injury happens first.
I think that's probably true. If you take something like, PART, primary age-related tauopathy, by definition, cases of PART have no beta amyloid deposits. That's how it's defined. And the tau assemblies are found localized, largely in the limbic region, so the anterolateral cortex and the hippocampus. And in our hands, the structures are identical in those from, let's say, Alzheimer filaments from frontal cortex. We have lots of beta amyloid deposits. So I think what this shows is that the tau fold of the Alzheimer tau fold can form in the absence of beta amyloid deposits. Perhaps when there are such deposits, there's more of it, I don't know, or maybe there are different mechanisms that beta amyloid deposits can also induce tau filaments, but at least I think the work we have done is consistent with the idea that primary age-related tauopathy doesn't prove it, but is a sort of component, I would call it, of early Alzheimer's disease. It certainly suggests, I think, that the pathology starts. It's consistent, at least, with the Bragg staging, the pathology starting in the limbic region and then sort of spreading to the cerebral cortex and in all the other diseases as well. I mean, one question, for instance, is corticobasal degeneration. There you have an abundant glial cell component of tau assemblies, and the question people often ask, is the filament structure the same in glial cells and in nerve cells? We haven't really looked at this specifically, but I would have thought that in the tissues we looked at of corticobasal degeneration, there were lots of glial cell inclusions. So we only ever saw that particular protofilament fold, nothing else. So,
thank you very much. Thank you.
Christian, is your turn.
Okay, I also had a question to Michel, and that is on translation in principle. So did you start looking also at aggregates from human, for example, overexpressed or mildly overexpressed tau also, and in order to see whether in mice they look the same as in humans?
Yeah.
And would you dare to say that if they look differently, that then translation doesn't work?
Yes.
From to human.
Yes, I think that's, that we have actually. Now for tau it's complicated because in mice you don't get any tau inclusions with wild type protein. It just doesn't work. If you overexpress it, it doesn't assemble into filaments. What you have to do is use a mutation in the tau gene that causes frontotemporal dementia in humans. So we have these mouse lines that overexpresses P301S tau, which is one of those inclusions. And we have determined the cryo-EM structures of those filaments. And it's not published, but it's basically finished. And the structures are different, clearly different from what you see in human brains. I mean, the caveat here is, though, that we don't know what the structures of tau filaments from humans with a P301S mutation, what those structures are, because there's no frozen tissue. And we are at the moment, because of this, looking at humans with a P301L mutation, of which there is tissue. And there is one case of a mutation called a P301T mutation from Spain, that we're also looking at, but we don't have those results yet. But it's clearly true that the mouse filaments, the structures are very different from those we have so far seen in human brains. And you may be right. I mean, that may be a reason. So, I mean, if you take my view on all this, I would have to argue that certainly these mouse tau filaments are probably not representative of what's happening in human brain.
But would that also then mean that one should go to other species, or?
I think what one should,
would that trash the whole work?
I have no idea. I mean, it's hard to say. But what I would think is that what one should first do try and find methods by which one can produce tau filaments in vitro that are like those from human brain, and then produce, if you have that, then produce transgenic mouse models of that and see whether that works better. I think different species, I don't know. I mean, what species would you use? And in the end, you end up having, you know, I don't know. In the end, you have using species that live longer than we do. So I don't know. I mean, I don't think one should do that.
Okay.
So I saw a question from Carmelo. Carmelo, it's your turn.
Can you talk?
Yes, yes.
Oh, I have a question for Bean. My question is a general question. So, when you find amyloid inhibitor, we think in our mind what model of I don't know, toxicity and so on, we consider. As you well understand, you use only the amyloid hypothesis. So, so you consider that all the amyloid is dangerous for cell. Why do you not consider the interaction of amylogenic proteins, so EAPP or E-beta or alpha-synuclein with membrane? Consider also the toxic amyloid hypothesis. Because if you want to find a target, we consider the path of toxicity. And I'm sure that the toxicity, the origin is the membrane of the cell. What do you think about this question?
Thank you for your question.
I understand it's a hard question, yes.
Thank you for your question. I think what I'm getting is the important role of membrane you are mentioning. We have not studied that, the aspects. I know there is a numerous important study, including Ram's lab, talking about the importance of the membrane for related to the amyloid-induced toxicity. We have not looked at those aspects.
Okay.
But there are some studies we have done related to the interaction of the inhibitor to the membrane, some of the membrane lipid related to lipid bilayer, and we have not going more extensively, and we have not in this talk have not discussed on that. But I think membrane is important, dimension related to the cytotoxicity of in general of those amyloid, and I think probably will be related to the inhibitor as well.
Okay, thanks.
Thank you.
So Omar just joined us. So Omar, you can go ahead and ask your question. You're muted.
Sorry. Thank you, Magdalene. Thank you, Michel, for the fantastic presentation. I just have a few questions for you. The first question, which is the filaments you isolated from the MSA, the lacking the C terminal. As you know, the immunohistochemistry, we use antiphospho at selenium-129 to stain the glial cytoplasmic inclusions. So how can we explain that? I mean, the missing part, which is you don't see in the filaments you characterized. The second question is, if we take the filaments you isolated and you inject it to the brain, and I know the Brusners and others injected, and do you think they will replicate what we have seen in the MSA brains, or they have shown that they didn't see that. How can we explain that? And what's the best way forward to establish model or animal model?
Yeah, so the first question, the C-terminus of alpha-synuclein is not present in the core of the filaments, but we don't know whether the C-terminus was present in the filaments. I mean, if you look at the tau filaments, this is more striking. I mean, they have this huge, most of the molecule is fuzzy coat. So we don't see that. Certainly in the particular cases of MSA that we used for the structural work, but this is just by histology. I don't think we've done by immuno-EM. Certainly, many inclusions are positive for phosphorylated serine 129. So I suspect, I would have thought that it was probably that the C-terminal region was probably there in the majority, at least, of those filaments. Now, for the second question, we have done some experiments where we used sarcosyl insoluble fractions, which contains alpha-synuclein filaments, from at least, I think, only one of those MSA cases, and injected it into this mouse line that also Prusiner uses, called the M83 line, which is transgenic for A53T alpha-synuclein. In the heterozygous state, doesn't develop any pathology. And when you inject that induces assembly. But when you use, and we agree with him on that, when you use extracts from Parkinson's disease brains, for instance, you get it works much less well than for MSA, which would suggest that the structures might be different.
Thank you.
So there were a bunch of questions, Michel. I don't know if you had the time to look through them. I have one which I think it's kind of like you didn't get asked, I hope. And this is from Roberto Simon, who is saying great talk. And I have a question regarding the extra negativity charge hydrophilic density. Do you think this may fit an RNA molecule in a small molecule? And it's kind of, have you seen any other molecules coordinating in your fibers, which you extract?
Yeah, no, we have not. I mean, we've looked at that in terms of size, in terms of nucleic acids, RNA or DNA, and the density is probably too small for that. I don't think it's likely, but it could be. We don't know what it is, obviously, but it could. It's probably, I would say, it's probably between an ion and RNA, and I have no idea what it is, but it's something that we're working on, but mostly for chronic traumatic encephalopathy at the moment, which is a hydrophobic, right? It's not a hydrophilic one. But I think it would be very important to understand what these things are made of, because they could be crucial for the assemblies of these particular, to form these particular folds. On the other hand, they could also just be something that happens afterwards. But I'm afraid I have no idea. I don't know what they're made of.
Yeah, thank you. So anybody else? We're kind of trying to wrap up. It has been kind of a long session. Anybody else from?
Marcus has a question.
Oh, Marcus, sorry.
No problem. I actually have a question for Binju. So, I mean, you showed the 700 compounds which you screened from the NIH library. And I mean, but you only basically showed rosmarinic acid and EGCG. So were there any compounds which in the library which had a strong effect besides, let's say, these very well-known molecules? Right, I mean, you had this which you showed in red, which showed a strong effect on aggregation, right? But you effectively only discussed bicalin, rosmarinic acid, and stuff which is well known to inhibit in a rather unspecific manner the aggregation. So I was wondering if any of the compounds which from the 700 was more specific, or?
Yeah, so thank you for that question. I know this class of a catechol-containing compound, there are promiscuity because they potentially can have this general chemistry happening conjugate to the peptide, which can potentially, in theory, can conjugate to any protein. But those compounds are a lot happen in the nature, and we are consumed in our diet already. So potentially those compounds have a nutraceutical value. For the higher standard of drug discovery, we are looking for larger library screening to help to identify more novel scaffold. And we are also based on the scaffold of rosmarinic acid and design hopefully more specific inhibitor, as you mentioned. And that is another question, similar questions. So we do, there are several, so the catechol and acetylquinone, these class of groups are most evident from the library, so has a large number in the NIH drug repurposing library. We do have other class of compounds, as I mentioned, one, we find several related to a tetracycline-based molecules, as well as others. We are still studying those molecules to verify them and to see whether they have a ex vivo or in vivo efficacy.
Okay, thank you.
Thank you.
I think Christian has a question.
Yeah.
Christian. Oh, it's a mistake. There is a question there's a question by Volga Gursky in the chat box for Michael.
Oh, I missed that.
Say, a superb presentation. I have a question about the extra density that you see in the hydrophobic cavity or adjacent to the basic residue. Does it run as a continuous density stretch along the fibril axis like that for the PET ligand?
Yes, the answer is yes, it does. So the we believe that the amount of this material must be quite large, the trouble, the problem is finding out. In principle, it should be easy to find out what it's made of, but the problem is that these filaments, the sarcosyl extracted filaments we use, they're not completely pure. So there are all sorts of things in there. And so what we're doing is we're using Alzheimer's disease filaments sort of as a control, because they're structurally very similar to the CTE ones, but they don't have this density. And then we're comparing Alzheimer's disease with chronic traumatic encephalopathy to see whether we get differences. And we do get differences, but so far they don't really make much sense in terms of this hydrophobic density. So yeah, in terms of chronic traumatic encephalopathy, I mean, this is just my guess, but I would have thought it's probably going to be a small lipid or something like that. But obviously we have to find out what it is first before speculating too much.
Thank you very much to both the speakers. Fantastic talks. Sorry for the internet trouble that I had. I couldn't join you timely. Great talks, great talks. I loved it.
Yeah, thank you. Thank you very much. And thank you for staying this extra time. I really enjoyed your talks. And nice seeing you. It's always nice to see faces.
Thanks, Rams, for this fantastic series. Make our Saturday morning more productive and meaningful.
Thanks for your lovely participation. And we selected you purely based on participation, and we discovered you. This is one of the benefits of having a ZOOMinar.
Thanks
and
a lot.
glad to see you, Michael. Otherwise, I would be seeing we are in different areas, different sub areas, and ZOOMinar enabled.
Yeah, yes. We never met.
Yeah, great.
And it's even my birthday today.
Great, thank you.
Oh, happy birthday!
Oh, happy birthday!
Yeah, I spend that. Yes, now I can have a drink.
So hopefully it was a good birthday present then today.
Yeah, it was good. It was good.
All right.
Thanks.
Magda, shall we wind it up?
Yeah. So that's the end of it. Thank you so much for coming, and I'll see you next Saturday.
Thank you all very much. Thanks, Danilo. Thanks, Christian, and thanks, Camilo.
Thank you. Bye-bye.
Thanks, Magda.
Bye.
Bye.